Hediste diversicolor, more commonly known as the ragworm. This species, which is formally named Nereis diversicolor, is a polychaete worm composed of 90 to 120 segments. It can grow up to 12 centimeters in length and lives in U, Y or J-shaped burrows. It is found in the northeast Atlantic, with its distribution ranging from the Baltic Sea out to the Mediterranean and the Azores. More specifically, it inhabits sandy and muddy intertidal zones of beaches and estuaries because of a burrowing ability associated with these sediment types. Hediste diversicolor burrows in soft sediments to depths of around 15 centimeters to avoid predators and to escape from low temperature waters. This behaviour is important to the ecology of its habitat and plays a key role in organic matter mineralisation, nutrient regeneration and element cycling. This sediment destabilisation makes it easier for smaller myofauna to burrow into the sediment. Bioturbation of Hediste diversicolor allows deeper sediments to be ventilated and redistributes nutrients. In doing this, nutrients can be reduced much deeper than previously possible, increasing benthic respiration rates and stimulating sediment metabolism. Within their burrow, ragworms actively ventilate by performing a peristaltic and an undulatory motion. In doing so, they increase the rate at which water and suspended organic matter enter the burrows. This enrichment promotes microbial growth, which means that organic carbon and nitrogen can accumulate, creating a richer food source for deposit-feeding fauna. In fact, a study in 1981 by Reiser concluded that concentrations of nematodes and copepods increase around oxygen-rich ragworm burrows. Hediste diversicolor are unique in the fact they exhibit a wide range of feeding patterns. These include carnivory, scavenging, filter feeding on suspended particles and deposit feeding on the sediment. A study by Theodore in 1993 proved that in shallow waters, when the ragworms used filter feeding, the concentration of phytoplankton decreased. Ragworms predate on various seagrass species, which are very important in the ecosystems as they stabilise sediments. Their bioturbation further destabilises such sediments. Predation on smaller fauna is also common for ragworms. Their range of feeding types, in particular deposit feeding, are significant to ecosystem functioning. As such, ragworm feeding is considered a rate-limiting step in detritus processing in estuaries. It is also suggested that the feeding type expressed by this species corresponds to its life stage, with juveniles mainly exhibiting deposit feeding, whereas adults predate on larger organisms. This is mainly attributed to a change in their body size, since larger individuals consume more energy and therefore require a better source of energy. Diversicolor are predated by a wide range of animals, both benthic and pelagic, including fish, mollusks, crustaceans and birds. Often, predation is considered the limiting factor in ragworm populations, hence they are thought to be a key prey species to many organisms within their ecosystem. Within sediment there is competition between species. Against Neomysis integer, Hediste diversicolor is superior, most likely due to it being able to search for detritus at a faster rate and over a wider area than the mysid. As such, integer survival rates suffer in the presence of the ragworm. More recently, Hediste diversicolor has been shown to have a wide range of physiological detoxification methods. It is able to bind together toxic metals such as arsenic and lead using metallophenines. In doing so, it produces insoluble lysosomes, effectively detoxifying sediments and allowing less tolerant species to populate environments which would previously have been inhospitable. Due to its diverse feeding behaviours and the redox opportunities created by burrows, diversicolor is considered to have a key functioning role in estuarine environments. Finally, because of the species' high abundance in muddy and sandy coasts, it is used in studies such as the National Programme of Ecotoxicology and the Nereis Park experiment to assess environmental quality.